A big hello to all my KCET aspirants. Children, I hope you're happy and you're healthy. So today, we are going to solve the previous year paper together, right? So this paper is of the year 2024. And if you're asking me, ma'am, what's the difficulty level? I'll say it's moderately difficult, not too difficult, not too easy as well. It would be actually easy for you if you're someone who's very much thorough with your NCERT. So if you're thorough with your NCERT, everything becomes easy in the case of biology, right? So yes, we will solve it together. We'll see how it works. If you can, try to solve questions before I do, okay? So whenever you see a question, pause the video and try to answer it by yourself and then match it with me. In that way, basically you are attempting a previous year paper and you're saving your time as well in that sense. Works? So are we ready? If you're ready, do let me know in the comment section that we are ready, ma'am. We are going to score full in biology, right? So starting off with the first question, that is, which among the following is used to treat emphysema? What is emphysema? It is a respiratory disorder, my dear children. Now, in human beings, you need a protein that is alpha-1 antitrypsin. What? Alpha-1 antitrypsin. The first thing that you need is alpha-1 antitrypsin. Okay, it is what? It's not a hormone. It is not a hormone. It's a protein. So human hormone 1 antitrypsin becomes wrong. Okay, and human interferon again becomes wrong. Human protein antitrypsin, yes. So if you're adding alpha-1 antitrypsin, it becomes more easy. I don't know if it's a typo error or things like that. Anyways, the answer for this question is alpha-1 antitrypsin, and this is the one. It is a protein. It's a human protein. Now human lactalbumin, it is alpha-lactalbumin. I think the font color I didn't change, which is why it didn't appear. So alpha-lactalbumin is what? It is a milk protein found in human beings, and this particular protein was made to be produced by a cow whose name was Rosie, my dear children. Remember that also. So that becomes a transgenic animal. A cow was made to produce what? Human protein, that is alpha-lactalbumin. So none of them are the ones that is used to treat emphysema. There is only one, and that is human protein, alpha-1 antitrypsin. Okay, and same thing, you took the gene for that, put it in some organism and made that organism produce this particular thing so that we can treat patients with emphysema. Works? So I hope you never forget. C becomes your answer for this question. Now, going ahead, homeostasis is a condition where the organisms, what do you mean by homeostasis? So basically when I say in my body there is homeostasis, okay, my body maintains homeostasis. That means basically I am having a constant internal environment. Is it like when I go to sun, all the things in my body becomes changed? No. When I go to some snowy area, does everything change? In that way, if it is happening that way, it will be reacting very badly. I would be having a very bad reaction to all the different atmospheres or environments that I'm going into. So my dear children, this process of maintaining a constant body environment, internal environment, okay, regardless of what is happening externally, is known as homeostasis. Homeostasis. Okay, so maintaining a constant internal environment and ever-changing external environment. Yes. Okay, do not maintain a constant internal, wrong. Change their internal environment according to their external environment, my dear children. No. Change their internal environment when external environment is constant. Array, that cannot ever happen, right? So basically, your answer becomes A. Okay, maintenance of what? When everything, okay, you should be just like that. Whatever happens, you should be constantly preparing for KCET. Okay, anything, earthquake, tsunami, anything could happen. Well, you should not, okay? You should try to save yourself first and then prepare for any exam. But I'm just saying in a funny way that, my dear children, you have to remember whatever happens outside, whatever the situations are, if these are something that you can probably control, so control them. Internally, you have to be prepared for one examination, and that is KCET as of now. Then we will be having, let's say, pleasure time later on. Yes? So A becomes your answer. I hope you understood this. Going ahead to the next question. Which of the following is not a parasitic adaptation? Now, whenever in a question, not, incorrect, correct thing comes, you have to underline. You are asked about which is not a parasitic adaptation. Otherwise, sometimes you would look for what is parasitic adaptation. So, loss of unnecessary sense organs, that is a parasitic adaptation. Parasites do not need much organs, otherwise it will be very big, it won't be able to survive inside our body. It has to be really small, and for being compact, it removes all the unnecessary sense organs out there. So this is a feature of parasite, so cannot take it as not. Now, absence of an adhesive organ or suckers. Now, my dear children, these poor parasites which are staying inside our body, they cannot just stay inside our body like that. They need some suckers as well as adhesive substance so that they can stay, they can stick Okay, they are not poor by the way. Yeah, but yeah, they have to stick. So absence of adhesive organs or suckers is a wrong statement. Presence would be there. So which is not a parasitic feature? That is this. Absence of adhesive organs or suckers. Now loss of digestive system, yes, it is there. In parasites, they lose the digestive system also. There is high reproductive capacity. Obviously, they want to increase the number and cause more trouble to human beings, right? So yes, that is also parasitic adaptation. So all these cruel things that you see are parasitic adaptation. One which is not much cruel, that is absence of adhesive organs or suckers. That is basically the one which is not a parasitic adaptation. And that's my hair doing some fun things out there. Just give me a second. Where is it? Works? Chalo. Going ahead to the next question. Fourth, DNA polymerase of Thermus aquaticus is. DNA polymerase of Thermus aquaticus is what? What is Thermus aquaticus? It is a bacteria that stays at higher temperature. Now, if it is staying at higher temperature, probably it will have all the mechanism to stay in that particular environment. Yes, to combat. That is why it is staying there, right? Now, basically, suppose you just understand, if we do not have sweater, can we survive in colder regions? No. Only if we have sweater, we would be able to survive with that. Similarly, in Thermus aquaticus, it is able to survive because all of its bodily function is adapted to that. So, even my dear children, the DNA polymerase, which is responsible for replication of DNA, that is also able to survive at higher temperature. So what do you call this condition when they are able to survive at higher temperature? When they are stable, you call this as thermostable. What do you call this? Thermostable. So DNA polymerase of Thermus aquaticus is thermolabile? No. Thermophobic? No. Exonuclease? What are you talking about? It is thermostable. It will be stable at higher temperature. And this polymerase, which you get from Thermus aquaticus, is used in PCR, polymerase chain reaction, and you call this as TAC polymerase. Now you know, I suppose you know how the TAC name come from. So T for Thermus and AQ for aquaticus. Okay? Cool. Now, if a recombinant DNA bearing gene for resistance to ampicillin is transferred into E. coli cells. So my dear children, this is an E. coli cell. And over here, what is the question? If a recombinant DNA bearing gene for resistance. So this is the recombinant DNA that is bearing the gene for resistance to ampicillin. Ampicillin is what? A drug, an antibiotic. Now, host cells become transformed into ampicillin resistant cells. Don't you think that this particular E. coli can now survive in ampicillin medium? It becomes resistant to ampicillin because it has what? Ampicillin resistance gene. So even if I put the drug ampicillin, it won't still die because it is resistant to it because it has the gene. Now what happens when these E. coli are grown in a medium containing ampicillin? I've already explained you that. When these guys, okay, who have got the recombinant DNA containing the gene, if they are put in ampicillin medium, my dear children, will they survive or not? Will they survive or not? They will be surviving. Okay, they will be surviving, obviously. So what do you call this? When someone has taken up something new from outside, what do you call this? Can I call them transformants? Can I call these E. coli as what? Transformants? They have got transformed. They got something new, they have got transformed. Okay. Now, what happens when these E. coli are grown on medium containing ampicillin? You have to tell me. Non-transformants will grow and transformants will die. That is, the one that is having the recombinant DNA, they will die, and the non-transformants, as in the one that does not have this, okay, does not have this ampicillin resistant gene or nothing, okay, they will be growing. Do you think so? That can happen? Well, I don't think so. It's a wrong statement. Non-transformants will die. That means the one that do not have this resistant gene will be dying, and transformants will be growing. Why? Because yes, they have the gene for resistance. So this is a correct statement. Yes. Now both non-transformant transformants will die. Array. Both non-transformant transformant will grow. Wrong. Okay. So it is just B that becomes your answer. Works. Fifth question. All clear? Yes. Chalo. Children, do let me know if you're enjoying the class or it's like pretty boring, ma'am. You have to improve a lot. You have to tell me that. Now, question number 6. Which of the following is based upon the principle of antigen-antibody interaction? My dear children, what are antigen and what are antibodies? You should know that first. So, antigen is something foreign that enters our body. Anything foreign that enters our body which has the capacity to excite our immune response. Okay, to excite what? An immune response is known as an antigen. Fine. A dust particle can also be an antigen, because when dust particle gets inside, my immune system becomes activated. I will be doing achi. Right. Right. So this is a response that I am having. So basically, in response to antigen that enters our body is going to produce certain antibodies. Our body is going to produce what? Antibodies, my dear children, which will be basically targeting them and helping to kill them. These antigens, destroy these antigens so that they are not there. Okay. Now, which of this processes PCR, that is polymerase chain reaction, ELISA, that is enzyme linked immunosorbent assay, which is used for HIV test, our DNA technology, that is recombinant DNA technology, gel electrophoresis, my dear children, which of these do you think is having a principle of, is working on the principle of antigen antibody interaction? PCR basically is amplification of DNA. You want to increase the number of DNA, you do PCR. ELISA is basically used to find whether a person has HIV or not. Recombinant DNA technology is used for what? Creating new combinations of DNA which will help, get the products that we are looking out for. Gel electrophoresis is something, is a process through which you separate the DNA fragments along with their, fragment size. Right. So, if you talk about ELISA, this is the one. I'll tell you why this is the one, okay, so that you remember it forever. Now, in ELISA, you have different types of ELISA, my dear children. One type of ELISA is basically where you are going to put out the antibodies like this, okay. There will be antibodies like this, and you already know that these are the antibodies released in the body of people who are infected from HIV. Who are infected with HIV in their body, these antibodies are released. So, I know that. So, I synthetically made these antibodies and I have kept it over there. Now, I have got a blood sample of a person who has HIV, okay, who has HIV. I am going to take that and I am going to take the antigens being put down over here. If the antigen comes and binds on these antibodies, I'll get to know that, hey, these antigens got bounded on these antibodies only because probably they are there for killing it. Yeah, they are specific to each other, which is why it is coming and binding. But still, can we see it? Can we see that this binding has taken place? No. For that, we have another antibodies. Okay, we have another antibodies, and you know what the best part about these antibodies are? They are actually linked to, okay, they are actually linked to one enzyme. They are actually linked to one enzyme. So basically, when these guys come, they see that, hey, the same antigen is there. I have to go kill it. So it goes and traps it like this. It goes and binds over there, and basically you have the enzyme also. You have the enzyme also. Now, what is the use of enzyme, ma'am? Basically, now what you're going to do, you're going to put some substrate. You're going to put some substrate, and if the enzyme, and if this particular reaction has taken place, that is antibody have bonded with antigen, what will happen? Basically, the enzyme will be working on the substrate and giving a change in the whole thing out there. This is known as enzyme linked immunosorbent assay, and then you get to know that the person has HIV or not. You have different types of ELISA, my dear children. There is sandwich ELISA, there is this ELISA, that ELISA, and things like that. You do not need to go very deep, but just understand it is based on the principle of antigen antibody binding. Okay, I hope you never forget that. Fine, and I'll tell you one more answer with this. PCR. PCR is used to, PCR is a technique that we use to find if the amount of pathogen is in very less amount. With the help of PCR, we would be able to still find that this particular person has this disease. Why? Because in PCR, you are amplifying the content. So, even if little amount of pathogen is there, their genetic material is there, it will be able to amplify it and let us know that, hey, you are infected with this particular disease. So, please remember that goes for PCR. PCR is not just for amplification of DNA, but also for finding out a disease if it is there in trace amount in our body. Okay, please remember that. Now, a strict protection of biodiversity hotspots could reduce the ongoing mass extinction by almost. Now, how will you get to know the answer? Only if you have read NCERT. That's the straight thing. Okay, there is no conceptual thing out there, right? So, basically, if I am protecting the biodiversity hotspots strictly, this can reduce the ongoing mass extinction by almost how much percentage? 20%, 25%, 30%, 35%. What do you think so? What do you think so? The answer is 30%. Straight question. There is no need for explanation. Nothing. How can I explain? 20, 25, 35, how can I explain about that, right? So, it's just 30, you have to remember it. Okay? Works. Chalo. Next. Identify the incorrect match with respect to recently extinct animals and their place of extinction according to IUCN Red List. Okay. So, we have to see if they are matching. So dodo is from Mauritius, is it correct? Yes, it is correct. Quagga is from Africa. Yes, ma'am, it is correct. Thylacine is from Australia. Yes, ma'am, that is also correct. What about Stellar sea cow? Stellar sea cow, I will never forget it, because you know what, Stellar sea cow was a very pretty organism which was there, okay. Why? It was actually an aquatic organism, found in the sea obviously, but it looked like a cow. So imagine a cow not being in the place where it should be, okay. So it was like that, and it was one organism which was killed because of over-exploitation. What, my dear children? Over-exploitation. So there are two organisms which were killed, which became extinct because of over-exploitation. Please remember it. So these guys are the one that got extinct, right? But it has not been mentioned that whether they have been extinct because of over-exploitation or not. But there are two names that you have to remember. One is Stellar sea cow, and the next one is basically passenger pigeon. Passenger pigeon. Please remember these names as well, okay. Now, Stellar sea cow, is it from North America? No, it is from Russia. It is from Russia. So this is a wrong thing. Again, basics of NCERT clear? Then the answer is clear. But I am telling you, this question has been asked in different formats. So I will recommend you to go through this entire slide that I have given over here. Okay, very important. Now, moving ahead to the next thing. According to hypothesis proposed by environmental biologists, a relatively constant environment in tropics promotes what? So basically, you know, tropic region are there, temperate region are there. Temperate region are something that does not have a constant environment. Because of frequent glaciation and things like that, what happens, for example, I am an organism staying up there. I feel that, oh, finally it's over, it's all snowy and nice. But then all of a sudden glaciation happens, I die. Okay, so that's not a constant environment, which is why organisms do not have a time to specialize in their niche, in the place they are living, okay, and there is no much species diversity in the temperate regions. But in the tropic regions, where the environment is kind of more constant, okay, more constant in comparison to temperate, what will happen? That is the question. It will promote what? Niche specialization and lesser species diversity. Okay, niche specialization, yes, they have that, okay, they are specializing in the niche, that is there, but lesser species diversity is a wrong thing. Niche specialization and greater species diversity, obviously, yes, you are getting a constant environment, okay, you are giving a constant environment, all the organism would be flourishing out there, yeah, there will be more species diversity. Now, niche diversity and lesser species specialization, wrong. Niche diversity and greater species specialization, wrong. Niche specialization and greater species diversity, this becomes your answer. 9th, B. Okay, next, in the prevention of air pollution, the role of scrubber is to remove. Now, this is a deleted portion question. Okay, but still I can tell you the answer for this. So there is a device that is known as scrubber, and what does the scrubber do? It is containing liquid, my dear children. Okay, it contains some liquid which basically captures the gaseous pollutants out there. Okay, it captures what? The gaseous pollutants out there. So in the prevention of air pollution, the role of scrubber is to remove what? Remove what? Particulate SO2. Actually, that is also being removed, but still, liquid SO2, gaseous SO2, liquid SO3, the answer becomes what? Gaseous SO2. Okay, guys, just give me a second out there. Yes. Now, going ahead with the next question. Match list 1 with list 2 and choose the correct answer. Okay, let's start again. I think almost in the deleted portion. So let's see. Nitrogen rich fertilizers, what do they do? Carbon dioxide, what do they do? Carbon monoxide, what do they do? CFCs, chlorofluorocarbons, what do they do? So basically, according to, even if I didn't study 11th and 12th properly, with what I studied in 10th grade, I can try to find answer for this question. I'll tell you why. CFCs, chlorofluorocarbons, all of you studied, they pose a big threat to what? The ozone layer. They kind of destroy the O3 thing. Okay, you know what? O3 molecules are the one that basically makes up the ozone, right? That is known as ozone molecule, right? So basically, CFCs goes and tries to break this thing up. So there is a breakup happening because of CFCs. So I'll say CFCs directly targets, why am I choosing different colors? Okay, CFCs directly targets what? Ozone, and causes depletion in there. And CFCs were being produced from refrigerators. So 4 goes with P. So P with 4, I just have one option, and with that I can get the answer for this question. Okay, just because I knew one thing. Now, nitrogen-rich fertilizers. So, nitrogen-rich fertilizers, my dear children, when you put on the plants, from the soil, it will be it will be going into what? The water areas. Now, in water, because there is enough amount of nitrogen, the algae which is there, it will start blooming, my dear children. Because there is so much amount of nutrient content there, it will start blooming. And this is known as eutrophication. What do you call this? Eutrophication. Basically, when the amount of content in there, in the water, the nutrient content, it increases way too much, which is why algal blooms are being caused. Okay, so that is eutrophication. So this becomes your answer for this. Now, carbon dioxide, what is it? What are we left with? Greenhouse effect and air pollutant, my dear children. Carbon dioxide, we all know it causes the greenhouse effect. So again, this goes with this. Now, carbon monoxide, it is a popular air pollutant. Carbon monoxide, you know what? If we are staying in an environment with carbon monoxide has higher affinity to oxygen than oxygen. Sorry, higher affinity to hemoglobin than oxygen. So it will go bind there, and then we got. Okay. So carbon monoxide, air pollutant, and with that we get the answer, that is B. Now, going ahead with something that is off in there on our syllabus, that is following representation PQR denotes steps of Griffith experiment. I hope you're aware about Griffith experiment. He was he was wanting to find what is the genetic material. Okay, so identify the correct one. R strain inject into mice die. So please let me tell you the story first. You try to find the answer for this. Until then, I'll tell you the story. So we have two strains of a bacteria, that's pneumococcus. One is R strain, another one is S strain. S strain basically had one extra layer out of it, which is why it looked smooth. Which is why it was smooth, my dear children. Okay, so these are the two strains of what? These are the two strains of a bacteria, that is pneumococcus. Now, when you're putting it in a mice, okay, when you're putting it in a mice, what happens is that when you're putting R strain, okay, live R strain, when you're putting in a mice, the mice will be happily living. Okay, it will live. But when you put an S strain, live S strain, the mice will, what happen? It will be dying, my dear children. It will be dying. Cool. Now, then basically Griffith thought that what if I am going to heat kill. Let's heat kill the S strain. Let's heat kill the S strain, and then that means try to treat it with heat and then put it in the mice. What will happen? The mice lives. The mice lives. Okay. So probably while heat killing, there was something happening, some changes happening in the genetic material, which is why it was not able to do the effect, and which is why the mice was able to live. Okay. Griffith was still not happy. Griffith said, I want to do one more thing. Let me take R strain, live R strain, which was not causing any trouble, and let me take heat killed S strain. Now let me put it together. Let's see what will happen. Again, when this was put, what happened, my dear children? Something strange happened. So heat killed S strain alone was letting the mice live. R strain was also letting the mice live. But when you're putting it together, the mice was what? Dying. So probably it happened because from this heat killed S strain, something was getting transferred to the R strain, okay, making the R strain convert into what? An S strain. An S strain. So basically, something was being passed which transformed the R strain into what? S strain, killing the mice, my dear children. Okay, this was the whole funda. So he was trying to find out what is that thing that is getting transferred. Was he able to find it out? No. He went ahead. Okay, there were other people, Avery, MacLeod and McCarthy, who said that it is the DNA which is the genetic material. But no one believed them. So again, Hershey and Chase did their experiment also, and finally it was proved that DNA is the genetic material, my dear children. So you should be knowing what all things were being used for proving that. Okay, I just spoke about Griffith experiment. Go read the other experiments as well. Very, very important. So R strain, when we inject into mice, does the mice die? No, Baba, it lives. It will be living. So this is wrong. Now S strain, heat killed one. Heat killed. If I inject into mice, the mice dies? No, mice will be living. Okay. Now R strain, I am injecting into the mice will live? Yes, my dear children. Always remember, something that appears smooth and shiny is not always good. So that is the one that causes trouble. So only if you put S strain, okay, the mice will be dying, or if you put heat killed S strain and also R strain together, the mice will be dying. So I'll say only this statement is correct, rest all of them are false. So R only, I just have one thing, that is B. So B becomes the correct answer for this particular question. Okay. Now, going ahead with the next thing. In tRNA, the region that binds with mRNA is called, in tRNA, my dear children, if you see tRNA, the region, so the tRNA is having 5' and 3' end, okay. So, this is an anticodon loop, anticodon loop. Suppose this is the mRNA which is in 5' to 3' direction, and over here you have AUG. This is the codon, and something that will bind to this codon is there in the anticodon loop, okay. It will have the exact opposite of it. So, if it's A here, it will be having U there. If it's U here, it will be having A there. If it's G here, it will be having C there, okay. So, this is the 3' end, this is the 5' end, and what does the 3' end have? It has an amino acid also. Please remember that, okay. Now, in tRNA, this is the tRNA, the region that binds with mRNA is what? The anticodon loop of tRNA. Amino acid acceptor end of tRNA, that is this. Does it bind to the mRNA? No, my dear children. Amino acyl synthetase loop of tRNA, again the same thing. Ribosomal binding loop of tRNA, my dear children, there is no something like that. So, basically what becomes your answer? A becomes your answer, anticodon loop of tRNA. And also remember, the process of adding of amino acid is known as amino acylation or charging of tRNA. Okay, amino acylation or charging of tRNA, same thing. It happens in the 3' end. Remember that. In which direction does the translation happen? From here. If suppose this is the mRNA, the ribosome would be moving from 5' to 3' direction. Just like how DNA polymerase, RNA polymerase, everyone does that. Okay? Cool. Now, the mRNA has an untranslated region. Okay, so you saw this process. What is this process? This is known as translation, where you're translating the information, okay, of making up a protein right there, right? So you're putting amino acid and amino acid come together to form what? A protein. So from an mRNA, you're translating the information which is there in mRNA to make a protein out there. Now, there are certain untranslated regions as well, something that does not undergo translation. Where are they located? So it is located in front of the promoter region as well as at the end over here after the terminator. Okay, so AUG is a start codon, it is the initiator codon. It initiates what? The process of translation. Okay, this is the first codon, always it will be like that. So you will be finding it towards this side of AUG, yeah, untranslated region, and also towards that side. Ma'am, why is it important? Because these untranslated regions which are there are really important for initiating the process of translation. Okay, it guides that, hey, you have to come and bind over here and things like that. So all this initiation, and for stabilizing also, stabilizing the mRNA which is undergoing translation, you have untranslated region, okay, before the AUG, that is at the 5' end, and also after the stop codons at the 3' end. So here you find stop codons in the 3' end, and after that you will be having the untranslated region, and here also you have that. Untranslated regions are also known as UTR. Okay, UTR. So mRNA has untranslated regions at 3' end beyond terminator codon. Do you think it is beyond terminator codon? At 3' end beyond terminator codon? Yes, it is correct. At 5' end before AUG? That is also correct. At both 3' end and 5' end beyond terminator codon and before AUG respectively? Yes, correct, right? At both 3' end and 5' end beyond terminator codon. At 3' end it's beyond terminator codon and it's before AUG at the 5' end. Now, my dear children, you have to be really careful. What is it to say? It is saying, if suppose it was 5' end over here, 3' end over there, then this whole thing would have been wrong. 3' end terminator codon, 5' end AUG, here beyond is written, here before is written. All these pieces of information are really important. So this becomes your answer. And both the areas it is present. Okay. Now AUG and terminator codon flanks the UTR. This becomes your answer. Okay. Now, in structural gene, the template DNA strand has nucleotide sequence of 3' A, T, G, C, and the list goes on. Find the correct and complementary nucleotide sequence on the coding strand, my dear children. So basically, suppose this is the DNA. Now DNA has two strands, my dear children. One strand which is there, it is known as the template strand. And the other strand which is there, it is known as the coding strand. Template strand is from direction 5' to 3'. Are you? Just a second. It is from the direction 3' to 5'. 3', are you? Again? Come on. Yes. 3' to 5' and coding is from 5' to 3'. Which is the one that undergoes the process of transcription, my dear children? Only the template strand. So always remember T for transcription, T for template strand. So this is the one that is going to undergo transcription. Okay, now what you have to find is, you have to find the sequence of the coding strand. The sequence of the template strand is given, you have to find the sequence of the coding strand. So in what direction should it be? It should be from 5' to 3' direction. Now try to find the complementary strand and I'll be right. Okay, so I hope you try to find out. Let's see. So my dear children, it's a DNA strand itself. So 5', A goes with what? T. T goes with what? A. G goes with what? C. C goes with G. A with T. T with A. G with C. C with G. A with T. Okay, until now we will try to find out. First thing, 5' to 3' should be the thing. So you have to cancel these options which does not say 5' to 3'. Okay, eliminate them. Okay, now you have to see TAC. TAC? No, ATG. So this is wrong. What is there? This is correct. Do not go around doing the whole thing. You might not need it. Okay, so look for polarity first and then go ahead with anything that you're having. Always remember DNA polymerase, RNA polymerase, everything makes up a new strand from 5' to 3' direction. So always template strand will be in the opposite direction. 3' to the 5'. Okay, and with that you will be able to solve this question. Very easy question. Okay, now going ahead. Read the following statement. Statement 1 says, all vertebrates develop a row of vestigial gill slits during embryonic stage. So vestigial gill slits is something that we do not need. So during embryonic development, embryonic stage, when we are in our mother's womb, my dear children, all of the vertebrates which are there, vertebrates as in fishes, aves, as in birds, okay, amphibians, reptiles, mammals, everyone, when in embryonic stage, we all develop vestigial gill slits. We all have gill slit in embryonic stage which goes off later on. Is that true or false? It is absolutely true. It is what? Absolutely true. And that's not all. We also have tails which goes off. Okay. Now statement 2 says, embryos always pass through the adult stages of other animals. Do you think when we are embryos, we pass through the adult stages? Have you become fishes completely inside the mother's womb? Have you become amphibians, frog inside the mother's womb? No, my dear children. Okay, we definitely have some features of the organism, as I said, the gill slits which are usually found in fishes and things like that, or tail, but it's not like we always pass through adult stages of other animals. That will be totally wrong to say. We do not pass through the adult stages of all the other animals out there. So this is a wrong statement. This is the correct statement. Statement 1 is correct, statement 2 is false. Statement 1 is correct, statement 2 is false. A becomes your answer. Cool. Next, which of the following exhibits haplo-diplontic life cycle? Haplo-diplontic life cycle, my dear children. Which one has both haploid life cycle, okay, and also diploid life cycle? Haploid means half the number of chromosomes. Diploid means double, right? 2n. 2n. That's what you mean. So you have fucus, you have chlamydomonas, you have gelidium, you have ectocarpus. So what would be your answer for this question? It is from your PU1, yeah, and the answer for this question is ectocarpus. It is the one that has haplo-diplontic life cycle. Okay, most of them that you see over here, either they have haploid life cycle or diplontic life cycle. Okay, not both of them. Okay, and gelidium has something else only. So ectocarpus is the best answer for this, which has both haploid and diplontic life cycle. D becomes your answer. Next, identify the phylum which shows the following characteristics. Okay, animals are exclusively marine, that means they stay in salt water. They are radially symmetrical, that means they can be cut into equal halves from any particular point, okay, which passes through the center. And they are diploblastic, that means they have 2 germinal layers. What are 2 germinal layers? In diploblastic animals, ectoderm and endoderm. Okay, so usually we have 3 germinal layers. So in usual organism, ectoderm, endoderm, and in middle there is a mesoderm. But in these organisms, they do not have mesoderm, instead they have mesoglia. So this is not a complete layer that you think of as a germinal layer, which is why you say they're having only 2 germ layers. Okay, that is ectoderm and endoderm. So they are diploblastic. That is what it said. So children, what are those organisms that are radially symmetrical, can be cut down from equal halves from any area, and also diploblastic? Always remember there are only 3 of them. One is your Cnidaria. Okay, one is your Cnidarians. Second one is Ctenophora, the bioluminescent ones and things like that, okay, which have the, you call them comb jellies as well because they have strands like that, combs around their body, okay. And the third one is the adult form of echinodermata, as in starfishes. Okay, starfishes. So basically who all? Cnidarians, as in you have your, what do you call, jellyfish. Yes, you can cut them into equal halves from any plane which passes through the center. Ctenophora, as I told you. And then this thing, that is adult form of echinoderms. Always remember, larval form of echinoderms are bilateral symmetrical. That means they can be cut into equal halves only through one plane. But once they become adult, they can be cut from any plane. For example, starfish. Okay, so and now they have to be diploblastic. So we get to know these three are the ones. So with radially symmetrical, these three are the ones. Now diploblastic. Who among them are diploblastic? Only two. Cnidarians are diploblastic and also Ctenophora. They are diploblastic. None of the others. So we just have two options to choose from. Now, body bears 8 external rows of ciliated comb plates which helps in locomotion. I told you, if something has comb plates, they are comb jellies. Basically, you're talking about what? You're talking about Ctenophora. Cnidarians do not have this. Cnidaria, if you remember, they have polyp stage, medusa stage and things like that, but not this. So this is your Ctenophora. I got to understand from this only, but let's go ahead. Digestion is both extracellular and intercellular. That means the food is taken inside the cell and broken down, and also it is broken down outside the cell. Now reproduction only by sexual modes. Reproduction is only by sexual modes. What is the answer? Coelenterata, okay, Coelenterata, okay, or mollusca, arthropoda or Ctenophora. Your answer becomes Ctenophora. Okay, cool. Now, when a flower has both stamens and carpels, it is described as, that means it has both androecium as well as gynoecium. It is in this condition. So this is the flower. Okay, you have the female part as well as the male part in the same flower. What do you call this flower as? You call this as bisexual. What do you call? Bisexual, having both the sexes together by 2. Okay, asexual does not have anything. Unisexual means it will just have either this or this. Okay, dioecious plants, you say. Okay, what do you mean by dioecious plants? That means one plant will be having only female flowers and the other plant will be having only male flowers. Male flowers as in male organs will be only there. These are dioecious plants. That is not applied over here. So you're talking about one flower having both the stamen as well as the carpel. So you call them bisexual flowers. Okay, chalo. Now, ciliated epithelial cells are present in kidneys, intestine, blood vessels or bronchioles. Ciliated, cilia, you know, smokers, when someone smokes so badly, what happens? Their cilia is getting damaged. Why? Because, my dear, it's the respiratory tract that gets damaged, and the respiratory tract, you have cilia so that it can trap things out. So, ciliated epithelial cells are found in the bronchioles because that is the only respiratory part over here. Kidneys have different thing. Intestine have microvilli and things like that. Okay, now blood vessels have a whole different thing. Okay, over here you just have this thing. Blood vessels have squamous, kidneys have cuboidal and things like that. Okay, so and intestine has columnar epithelial cells and they have microvilli out there. So bronchioles are the one that have ciliated epithelial cells out there. They are also columnar, but they are ciliated. Okay, they have cilia. So, respiratory tract is something that you have to look forward. So, yes children, only Jasmine ma'am can do this. Okay. So, question number 21. Which of the following statement is correct with reference to vacuoles? Vacuoles are the big organelles that you find both in animal cell and plant cell. But in plant cell, it becomes more prominent. They occupy almost 90% of the whole plant cell. Yeah. So, it is membrane bound and contains storage proteins and lipids, my dear children. Can you tell me about that? Does it contain, it is a membrane bound structure? Are you sure about that? Yes. Yes, ma'am. They have a membrane and that is known as tonoplast. What do you call this? The membrane is known as tonoplast. Fine. Now, do they contain or they store proteins and lipids? I think that is a function of what do you call plastids. In plastid, you remember you have leucoplast, the colorless plastids, and the other one therefore storage of the fats, lipids and proteins and things like that. You have different names for that, right? So this is a wrong statement. Even the first part of it is correct. Now it is membrane bound and contains water and excretory substances. Yes, it definitely has, and this particular water which is there gives a structure to the plant cell as well. Okay, so this is correct. It lacks membrane and contains air. Array, how can it lack membrane, my dear children? Okay, it cannot lack membrane because it has tonoplast and contains air. Do you think it's floating? No. Okay, now it lacks membrane and contains water and excretory substances. Wrong. This statement is wrong. So what is the correct statement? You have B as the correct statement. Okay, going ahead. exoskeleton of arthropods is made up of unique complex polysaccharide known as polysaccharide, okay, carbohydrate, my dear children. So exoskeleton as in the outside skeleton, outside the body also they have a skeletonous structure, okay. What is it made up of? So if you have seen arthropods, cockroach and all, it's very difficult to kill them. Why? Because it is made up of hyaluronic acid. No, that we put on our face, right? Waxes, cellulose. Cellulose is there in all the plant cells which is there. The answer is chitin. What is it? Chitin. Remember it. Chitin. So even if we put a tin and try to kill the cockroach, it won't die. How's that trick? I know it's lame, but still. So chitin. Now the enzyme recombinase is required at which stage of meiosis 1? So meiosis 1, my dear children, is having prophase 1, metaphase 1, anaphase 1, telophase 1, right? Now in prophase 1, you have it divided into different phases. How do you remember it? Okay. Lazy zebras play during day. LZPDD. Okay. This is leptotene, this is zygotene, this is pichitin, this is diplotene, and this is dikinesis. The last one, di. Okay. So basically in leptotene, the chromosome and everything, what happens to them? Chromatin gets condensed. Okay, so chromosomes will be starting to get visible. So chromatin, when it condenses, it forms chromosome, which is why the chromosomes are going to be visible. Now slowly the other nucleolus and things like that, it is going to go off and things like that is starting off. Now in the case of zygotene, what happens? 2 homologous chromosomes, okay. So one chromosome, my dear children, suppose it is like this. One chromosome from each pair you get from each of your parent, one parent, second parent. Right. Now, in the phase before it, in the interphase, the chromosomes which were there, it has duplicated already. It has duplicated. So they also have a strand like this. So do not consider as this and this has 2 separate chromosomes, since they are joined together in one. Remember, my dear children, they are just duplicated form. Okay. So what do you call these chromosomes as? These 2 chromosomes, homologous chromosome. Why homologous chromosome, ma'am? Because this you got from your amma, this you got from your appa, right? Now over here, in this particular location, you will be having a gene coding for the height. Here also you will be having a gene coding for height. So these are known as homologous chromosome. Now these genes can be different. It can be capital T, this can be small t. That's not an issue. But at least they have same type of genes, okay, coding for the same character in what say at the same location. These are known as homologous chromosome. So homologous chromosomes come together and basically a gum is formed between them so that they stick together. This is known as synaptonemal complex. What do you call this? Synaptonemal complex, my dear children. Synapsis happens in the zygotene stage, Now in pichitin stage, what will happen? Pichitin, that is the best thing. In the pichitin, my dear children, slowly, you're very close to each other. They start, exchanging the stuff. They start giving food, thing like that. When we also become close, what will happen? We give each other a lot of food and whatever we like, right? So basically over here also, they are going to exchange certain things, and over here, this process is known as recombination, okay, or crossing over. What do you call this as? Crossing over. So this is a very important feature of pichitin stage. Crossing over happens over here. Okay, pichitin has a C, you can remember crossing over with C. So when crossing over happens, it cannot happen like randomly like that. You need an enzyme, and that enzyme is known as recombinase, which helps in recombination. So there will be crossing over like this. The chromosome goes off, and crossing over, forming an X-shaped structure over here. So this is the thing that you have to remember. Followed by, in diplotene stage, what will happen? Basically, the synaptonemal complex will go off. But do you think this will go off? No. Wherever they are linked together, you call this as chiasmata. What do you call this as? Chiasmata. So we say synaptonemal complex, it dissolutes, but the chromosomes stay together at position of chiasmata. Now in dikinesis, they are trying hard to break up, my dear children, but does not happen. Okay, this chiasmata, they try to pull each other, which is why chiasmata gets terminalized, as in it moves downwards, but it's still there. So terminalization of chiasmata happens over here, and the chromosome will be perfectly visible by this particular time. So this is prophase 1. And what is the question? The enzyme recombinase is required in which stage of meiosis 1? Your answer would be what? Pichitin. Pichitin, it has a C, C for crossing over. You can remember like that. I know I explained you further because you can get any question from anywhere. Okay, so I hope it is helpful for you. Now going ahead to the next question. The water potential of pure water is? My dear children, if it's pure water, the water potential would be 0. Okay, if this pure water has some solutes in it, it will be always negative. It cannot be positive, it will be always negative, less than 0. Cannot be more than 0, remember that. So water potential of pure water which does not have any solute, it is 0. Okay, if you are adding solutes into it, then it will be less than 0. Remember. Going ahead with the next question. Match the pigments given in list 1 with the color and chromatogram given in list 2. Okay, so I hope you are not able to see it. Okay, because even I cannot see it, so I'll zoom it in. Now you are able to see it? Yeah. So chlorophyll B, carotenoids, chlorophyll A, xanthophylls is there. So my dear children, I am pretty sure about one thing over here. Xanthophylls. Xanthophylls are the yellow pigments. Xanthophylls are what? The yellow pigments. So I'll say 4 goes with R. 4 goes with R. I am pretty sure about that. Okay. Now over here, 4 goes with R. Do I have an option like that? I have just one option, and with that I can find the answer. So this should be the answer. 1 with T. 1 with T. Let's see. Chlorophyll B gives yellow-green color. Chlorophyll B gives what? Yellow-green color. Carotenoids, my dear children. Carotenoids, it can be orangish red or yellow-orange. Let's see what is there over here, since that is the only option that is correct for it. So 4 goes with R, right? 4 goes with R. Yes, yes. So what do we have? 2 with P. That is, carotenoids is going with yellow-orange in this particular option. That's totally fine. Can go with this or this also. Now chlorophyll A. It is what giving the blue-green color. It's very much in the blue-green algae, you know, cyanobacteria. So chlorophyll A, my dear children, gives a blue-green color, whereas chlorophyll B gives a yellow-green color. Cool. And xanthophylls, yellow color. Carotenoids, either of them, but over here, since other options are matching this, so let's go with yellow-orange. Works. Xanthophylls yellow would be really easy for you to remember. Chalo. Next question. Which is the intermediate compound that links the end product of glycolysis with TCA cycle? So what is the end product of glycolysis, my dear children? You have the glucose breaking down into pyruvate. Now this pyruvate, this is happening in the cytoplasm. Now this pyruvate will be entering into what? This will be entering into what? The mitochondria. The mitochondria. Now in mitochondria, TCA cycle, citric acid cycle, or what do you call it as Krebs cycle would be taking place. It's the same thing, my dear children. And for that, we cannot use pyruvate. Pyruvate basically gets inside and gets converted to acetyl-CoA. What? Acetyl-CoA, which is a 2-carbon compound. This acetyl-CoA gets inside the Krebs cycle and does the whole thing. So what is the intermediate compound that links the end product of glycolysis with the TCA cycle? Is it acetyl-CoA, or is it pyruvic acid, or is it oxaloacetate, or is it citric acid? So what is the intermediate thing that comes in between? Will be acetyl-CoA, not pyruvic acid. It will be what? Acetyl-CoA, my dear children. Please remember, this is the common mistake that we do. That's why I am telling you. Okay? Answer is what? Acetyl-CoA. Works? Okay. Now, auxins are for apical dominance. Gibberellins are for what? Apical dominance, what do you mean by that? Basically, suppose this is a plant. It does not allow the lateral branches to come off, rather than it will make the plant grow towards the apex. Okay, that is known as apical dominance. So auxins are responsible for that. What about gibberellin? Does gibberellin give rise to adventitious shoot formation? What do you mean by adventitious shoot? Because normally shoots form like from the plumule, right? It goes up like that. Adventitious shoots means shoots coming off from different regions other than this, other than the normal region. That is not the thing. Accelerates abscission. Abscission means basically aging and things like that. Does it accelerate that? No. Closure of stomata? No, my dear children. Gibberellin is not responsible. It's the exact opposite of what you are saying. Bolting. Bolting is basically stem elongation, premature stem elongation. So bolting, and also in bolting you get elongation after the stem elongation. You also have flowering. There will be premature flowering and also stem elongation. So first of all, stem elongates, and then you have what? The flowering as well. This process is known as bolting, and gibberellin are pretty much famous for that. Okay, if you are asked in sugarcane what is the reason for stem elongation, it is gibberellin. Okay, always remember that. So D, bolting, becomes the correct answer for this particular question. Again, PU1 question. Now, the term uremia refers to what? Accumulation of urea in the blood, presence of glucose in the urine, accumulation of uric acid in the blood, accumulation of uric acid in the kidneys. The answer is straight. The answer is accumulation of urea in the blood. In the blood, when urea gets, let's say, deposited or accumulated, you call this condition as uremia. Cool? Works for you? Yes. Now, the typical lubbed up sounds heard during the heartbeats are produced due to what? the lub-dub sound that you get during heartbeats. What does it suggest? Closure of semilunar valves. What are semilunar valves, my dear children? Basically the valves which are present between the ventricle and the aorta, as well as between the right ventricle and the pulmonary artery. Okay, those are the ones. So I'll show you. Not to confuse you, if I said something wrong, I'll make it correct. Let me see. So this is the left atrium, left ventricle, right atrium, right ventricle. Okay. Now over here you have the aorta running. This is a semilunar valve. The aortic valve is a semilunar valve. Okay. Now over here, from here to here, when blood goes to back to the lungs also, back to the lungs, this valve is known as pulmonary valve, and this is also semilunar valve. Why do I call it semilunar valve, ma'am? Basically they have a half moon shape, that's why. Now in between the right atrium and the right ventricle also you have valves. Also you have valves here, also you have that. They are atrioventricular valves between the atrium and the ventricles. Okay. Okay, so the one that is there on the right side, it is known as tricuspid. It is known as tricuspid. Tri has right term in it, so it is in right area. Okay, and then you have bicuspid towards the left side. Works. So now you are asked, why do we get lub-dub sound? That is the question. So my dear children, you get the lub sound when in the atria the blood gets filled. Okay, here also you get some things. Okay, so blood gets filled. When this blood is going to move to the ventricle, suppose when it goes to the ventricle, now what happens? The ventricle gets filled. Ventricle has to pump this blood towards the entire body and also to the lungs. So it will contract. When it contracts, what is happening is that these walls are opened up like this. These are usually in the open state, putting down all the blood into the ventricles. Now when the ventricles are going to contract, what will happen? The blood which is there over here will be pushing this thing inside. So something which was like this becomes like this because of the pressure. And now this is known as lub sound. When it closes, okay, when the tricuspid and the bicuspid valves, they close off, this is the lub sound that you get. Now, what about the dub sound? Dub sound is basically when the blood went off. Suppose it was contracted, okay, the ventricles contracted. Imagine the blood is going off. So it is not in this direction usually, it's in the upper direction. So what happens is that there can be a backflow of blood. There can be a backflow of blood, which is why these two things have to be closed again. These are semilunar valves, they have to be closed. And that when that is closed, it is known as the dub sound. The dub sound. So first sound, lub, is because of these guys. The dub sound is because of semilunar valves. Fine. So this becomes your answer. So closure of semilunar valves alone is not the answer. Okay, and closure of bicuspid and tricuspid, no. Closure of bicuspid and tricuspid followed by semilunar valves, yes. Opening of bicuspid and tricuspid followed by semilunar, no children, it's closure. That is why you get dub sound. When you close the door like that, that's when you get the dub sound, right? Similarly over here also. Fine? Okay. Now the question again, to confuse you guys, they might say semilunar valves followed by this. You should be knowing what the term followed means. Okay, so this happens, followed by this means after this, this will be happening. The second part will be happening. Now, next, question number 30. The functional unit of contraction is a portion of myofibril between two successive Z lines, portion of myofibril between two successive M lines, and things like that. What is the answer for this particular question? Can you try guessing it out? So basically, suppose you have the actin. Okay, I have to make it a little far. I don't know how successful I would be, but yeah. And then you have the myosin. Okay, this is what? The myosin. Just a second, ma'am. Okay, so this area, this is the myosin band and this is the actin filament that you are having, right? So this area where you just have the myosin, you call this as M line. This line is M line, okay? And over here where you just have the actin, this will be known as the Z line. So here also you will be having something like that. This will be known as Z line. What will happen next to it? Again, you will be having the myosin filaments. So this area where you just have the actin, you have Z line, and that area is also Z line. In between, when you just have the M, it is known as myosin. You call this as M line. Now this thing that is coming between two Z lines, the whole thing that you see, this whole unit is known as a sarcomere. Is known as what? A sarcomere, the unit of contraction. So in the functional unit of contraction is what? Portion of myofibril between two successive Z lines. Will that be correct? Yes. Portion of myofibril is between two successive M lines. No, that would not be correct. This is the correct thing. Between two Z lines, this portion where you have both myofibril as well as actin, this becomes the correct. Now, center of each zone? No. Okay. Center of the I band? No. What is the I band? This is the I band where you just have the Z line, where we just have the actin. Okay. And also, H zone is this, where you just have the myosin. Okay, so your answer is this, and with that we complete 30 questions. I want you to go through all the questions and I will be right back with the 31st question. So I hope you revised, and I'm so sorry, Aishwarya ma'am came in, so I had to move out for something else. Yes, and I was explaining this, but if you still didn't get it, I have a picture for that as well. So over here, my dear children, as you see, this pink color bands are the myosin filaments that you have, and this one is the actin filaments that you have. Right. Now, over this area, if you see, this is a Z line, this is a Z line. In between that, you have the actin filaments and the myosin filaments. So, the area which has both of them, it is known as the A band. Okay. Now, in that, if you see the middle area where there is only myosin, you call this an H zone, and also there is a line, this is known as M line. Fine. Similarly, over here where you just have actin, you call this as I band. You call this as I band. Now, that could extend this side also. Similarly, it will be also I band. Now, in between these two things, okay, in between these two things, the whole thing that you see over here is sarcomere, where what happens when the contraction happens, no, they move like this. They move like this. So what happens? The Z line and the M line, it will become close. Don't you agree? Yes, the Z and the M line, it will be becoming close when it comes together. And also if you talk about the H zone, don't you think it will be highly reduced? Yes, it will be highly reduced. So these are the things that you should be knowing about what? About contraction. So functional unit of contraction is portion of myofibril between two successive Z lines. Fine. Now I hope I was able to explain better with the picture. Now match the parts of a brain given in list 1 with their functions given in list 2. So you have medulla oblongata, you have hypothalamus, you have cerebral cortex and the limbic system. Now one thing about medulla oblongata I tell in every single lecture, okay. So basically there was a movie in Malayalam where it's like there is a hit in medulla oblongata and the guy goes off, okay. So why? Because this is a very important part of our brain. It forms the brain stem, it is right over here, and basically it is responsible for the respiration rate, for the heartbeat rate, for all of these things. It's a very, very important thing. So medulla oblongata goes with respiration. Okay, medulla oblongata would be going with respiration. I can zoom it and show it to you. 1 goes with R. 1 goes with R. With that, can I find the answer? Yes. Just because I knew one option, I could find the whole answer. 1 goes with R. There are none of the other options which have it. Now, going ahead with hypothalamus. So, hypothalamus is basically responsible for all your physiological function. Your hunger, your thirst, the temperature, the sex drive, lot of things. Right. So, hypothalamus goes with body temperature. Okay, what does it go with? It goes with body temperature. Now, cerebral cortex is responsible for all your motor function, the involuntary functions writing around there. It is what? Cerebral cortex that you have. It is responsible for the motor functions as a movement and things like that. That need coordination, right? Now, limbic system is responsible for olfaction, for sensing the smell and things like that. Fine. So, medulla oblongata, respiration, hypothalamus, body temperature, cerebral cortex, motor function and limbic system is what? Olfaction. Now, going ahead, so what will be your answer for this particular question? Your answer would be D. Now, as I said, if you know one option, you can get the whole answer. Now, going ahead, Hydra reproduces asexually by producing what? So, if you have seen Hydra, my dear children, if you have seen Hydra, it's a very fun thing, very cute little fun thing that you have. So, it has tentacles like this. Okay, and basically it forms small hydras by forming buds. They have buds with the tentacles. So this process of forming buds and then later moving away from the parent body and developing by itself is known as budding. First of all, they form the bud and it grows in size, and after that this particular bud which is there, it dissociates from the body and then it grows independently. This is known as budding. So it's not zoospores, it's not conidia, it is just buds. It's a very easy thing you might have studied in your 10th grade as well, not Gemule as well. Okay? Now, When male gamete and the female gametes are morphologically distinct, the condition is known as, they do not look the similar morphologically, as in their appearance, the physical structure is, my dear children, different. What do you call them? Homogametes, heterogametes, hermaphrodites, sexual dimorphism. So homogametes means they look exactly the similar. Okay, two round things, just imagine. They are homogametes. Heterogametes, for example, in human case, we have ovum which is circular and sperm which is elongated. Do they seem similar to you morphologically? No. So they will be what? Hetero. Hetero means other. Gametes mean gametes. Okay. So heterogametes means they do not look the same. Now hermaphrodites are those organisms which have both the male and the female reproductive parts within them. They are hermaphrodites. So they do not necessarily depend on some other organism for fertilization, because in their body only the male and the female gametes are being released. Sexual dimorphism is basically when two sexes appear different. For example, male and the female appears different. Again in us, do all the boys and the girls look different, right? Do we look similar? No, we have morphological differences, my dear children, between male and the female gender, right? So this is known as sexual dimorphism. So over here that has not been asked. What has been asked? When the male and the female gametes are morphologically distinct, what is this condition known as? This condition is known as heterogametes. Okay, you call this as heterogametes. B is your answer. Now, the role of filiform apparatus in the synergid is to what? What is the role of filiform apparatus in the synergid? So over here, you have what? You have the embryo sac. In the embryo sac, you have the synergids. Just give me a second. Okay, and the synergids, my dear children, they have the filiform apparatus, and over here you have the egg cell. Okay, this is the egg cell that you are having, and they have the antipodal cells, and they have the polar nuclei. Now, why do you think there is a need of filiform apparatus over here? So, you know, pollen tube, it carries the male gametes and it comes over here. It comes over here, right? So, there should be someone who could be guiding this pollen grain into the embryo sac towards the egg cell. And who is that? It is the filiform apparatus, okay, which is an appendage that comes in the synergids. So, role of filiform apparatus in the synergid is to what? Protect the egg apparatus. What is egg apparatus? This is the egg apparatus. The egg with the synergid is known as egg apparatus. It is not to protect the egg apparatus. Now going ahead. Endosperm formation? No, my dear children. Endosperm is formed. This basically I was told about two male gametes. One male gamete fuses with the egg cell to form the zygote. Another male gamete fuses with the two polar nuclei forming the endosperm. So that is not the role of filiform apparatus. Guide the entry of pollen tube? Yes, this is a role. It helps in guiding the entry of the pollen tube so that it can undergo later on, the male gametes can undergo fertilization. Prevention of gamete entry? No, that is not at all possible. So what is your answer? You just have one answer, and that is this. Guide the entry of pollen tube. Got it? Yes. Now, next question. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. So, same plant, another flower. So, this is one flower. This is another flower. What you are doing? Transfer of pollen grains from the anther to the stigma. So, this, suppose this is a flower. Okay, and then this is another flower. From here to here, from the anther to the stigma of different flowers but of the same plant, what do you call this process as? What do you call this transfer as? Xenogamy, autogamy, cleistogamy, guitonogamy. You call this condition as guitonogamy. This is cross-pollination. This is cross-pollination, but genetically if you see, it is just like autogamy. Why? Because it's the same plant. So there is no genetic variation coming up here. Okay, is there a genetic variation coming up here? No, my dear children. Same parent is producing both the gametes. So genetically it will be similar to autogamy, but it is cross-pollination, my dear children, and this is known as guitonogamy. When suppose in this particular flower, from here to here things are going, okay, in the same flower it is known as autogamy. Now from different flowers to this flower it is going, it is known as xenogamy. Cross, xeno, X is there, so cross is there. Okay, so for this your correct answer would be guitonogamy. Now, match the content of list 1 with list 2. What is the answer for this question? Polyombrani, perisperm, false fruit and parthenocarpy. Chalo. Tell me, can you tell me the answer for this? So over here, the most easiest thing is false fruit. Okay, most easiest thing is false fruit. False fruit is basically, you know what? Usually it's the ovary that becomes the fruit later on. Okay, after fertilization, it's the ovary that becomes the fruit. What do you mean by false fruit, my dear children? False fruit is basically when it's not just the ovary that is giving rise to a fruit, but the other part of the flower. For example, probably if the thalamus is being, there's a thalamus onto which all your petals and things like that are attached, right? Suppose this thalamus is also contributing for fruit formation. Okay, so over here, apart from ovary, something else is also there for fruit formation. This will be known as a false fruit. Okay, and the perfect example for this is apple. So can I say 3 goes with S? I can say that 3 goes with S. So let me try to find that out with just this option. Let me see if I am able to find the answer with this option. 3 with S, 3 with S. I have 3 options like that. So basically I cannot find out, but I can at least rule out one option, that is this. Okay, now the next thing that I am pretty sure about is perisperm. What is perisperm, my dear children? So usually in an embryo there is endosperm or you have cotyledon. Okay, basically you have either cotyledon or you have endosperm for nourishment of the embryo, right? Or you have both. So in some cases what happens is that there are certain cells that stays back. So suppose this is an ovule. Inside the ovule primarily we have the cells which are known as nucellus cells. Okay, nucellus cells. And one of the nucellus cell is the one that becomes the megaspore mother cell and undergoes division and forms the embryo sac and the female gamete and lot of things. Not male gamete, female gamete and lot of things. And usually this layer of nucellus, it goes off. But in some it persists. In some it will be persisting and it will be helping in the nourishment of the embryo which is there. So this persistent layer which is there, it is known as perisperm, and it is formed of the cells of the nucellus, my dear children. Always remember, okay? Perisperm and pericarp is different. Pericarp is the outer layer of the fruits, okay, or basically the ovary, what do you call it as. But over here, perisperm is the extra layer that you have got of the nucellus which does not degenerate and helps in nourishment. And it is found in 2 plants. One is black pepper and the other one is beet. So I'll say B square. I'll say B square. So over here, perisperm again. With this, can I try to find the answer? Perisperm with black pepper. 2 goes with P. 2 goes with P. I have 2 options, so I can eliminate one more option. Let me eliminate this option as well. Okay. Now, going ahead. Polyembryony. What do you mean by that? Polyembryony means you have multiple embryos being formed within a ovule, my dear children. This is known as polyembryony and this is found in citrus fruits. Okay, this is found in citrus fruits. So over here, where do you find which of the plant or what is left, which is citrus, my dear children? It's lemon. It's what? Lemon. So polyembryony would be seen in what? Lemon. So one goes with R, one goes with R, and this is what? This is how I can find the answer for the question as well. So A goes with this. Okay, 1 goes with what? R, my dear children. And what we are left with? Parthenocarpy. What do you mean by parthenocarpy? Parthenocarpy is basically when you are making fruits without fertilization. So as I told you, this is the ovary. Inside that you have the ovule. Ovule, when it undergoes fertilization, it becomes the seed, and ovary will become the fruit. But suppose fertilization has not happened. Suppose fertilization has not happened, still there is formation of fruit. Why? Because of some hormonal changes that is happening in the plant's body. This is known as parthenocarpy. This would be known as parthenocarpy, and you see that in banana. And since the fruit is forming without fertilization, so you do not expect seed to be there. So parthenocarpic fruits, if you see generally, they are seedless. They are what? Seedless. So this becomes your answer. I hope that is clear. Fine. Next question. Which of the following hormones is not secreted by human placenta? So human placenta only secretes 4 hormones. One is progesterone, estrogen, and then human chorionic gonadotropin and human placental lactogen. So HPL, and then what do you have? HCG, and then what do you have? What do you have? So I'm not writing in the form. Okay, anyways, HCG, and then you have progesterone, and then you have estrogen. So you see LS is luteinizing hormone, and luteinizing hormone is released by pituitary, not the placenta. Placenta only releases these hormones, please remember. And I remember in one of my live class, someone asked me difference between progestogen and progesterone. So progesterone is a natural human hormone that is produced in our body, and as I said that day, progesterone is a synthetic hormone, but we cannot completely categorize it under synthetic hormone as well, because progesterone is a combination, like it's a what do you call it, a group of hormones which is going to act like progesterone. Okay, so there can be natural version of it, there can be synthetic version of it as well. So either way, not to confuse you guys, we can in this particular level, in 12th standard level, we can use it interchangeably as well. You do not have to worry about that. Okay, so you can either say progesterone or progesterone, even though there are differences between them. Fine. Now, in human females, the endometrium of uterus consists of what? The endometrium, you know, uterus has 3 layers. The outermost layer is known as perimetrium, and then you have the muscular layer, which is known as myometrium, and then you have the innermost layer, which is glandular, my dear children, and this is known as endometrium. This is known as endometrium. So in human females, the endometrium of the uterus consists of smooth muscles. No, my dear children, it's the myometrium that consists of smooth muscles. Glandular layer, yes, endometrium is the glandular layer. Adipose layer, that is fat, my dear children, does not make sense over here. Cartilaginous layer does not make sense over here. So your answer, the endometrium of the uterus is what? The glandular layer, which will be helping in what? Implantation to happen. Okay, the embryo will be coming and implanting and later growing. Fine. So that is the answer. And please remember, myometrium, smooth muscles are involuntary. It's not under our control. We cannot say that, hey, you can do this, hey, you can do that. It will happen involuntarily. Now, going ahead to the next question. If 2 primary spermatocytes, remember, 2 primary spermatocytes, okay, and 2 primary oocytes undergo meiosis simultaneously, what will be the ratio of spermatozoa and ova produced at the end of gametogenesis? Always remember, from one primary oocyte you get 4 sperms. So from here you get 4, I'll show you how. From here also you get 4. But from one ova you get, from one oogonia you get from one primary oocyte you get 1 ovum. Similarly, you get 1 over here. So here it is 4, but here it is 1, 1. Here it is 1, 1. Okay, so 1 plus 1 is basically 2 and 4 plus 4 is basically 8. So we get a ratio that is 8:2. Okay, if I do like this, it is 4:1. This should be your answer, and I'll tell you how did I get this. So primary spermatocyte is there. It undergoes division, meiosis 1, 2 cells are formed. Then it undergoes division, 2 cells over here. This is meiosis 2. In total, how many cells are there? You get 4 cells, and 4 of them are the sperms that you're going to get. Similarly, same thing is going to happen over here. This is going to divide into 2. This is also going to divide into 2. This is dividing into 2. You get 4 cells. 4 plus 4, you get 8. Now, in the case of, so this is primary spermatocyte. This is secondary spermatocyte. This is spermatid transform into spermatozoa. Later you get sperm. Okay. Now, over here, in the case of primary oocyte, what is happening? It undergoes meiosis 1. It divides, gives rise to 2 cells. One cell is very small, one cell is very big. Okay. This small cell is known as first polar body. It does not divide further. You just have one cell which will undergo division. This is known as secondary oocyte. It will again divide, meiosis 2. Again a small cell and a big cell. This big cell that you get is ovum. So from one cell, how many cells did you get at the end? Only 1. Same thing happens over there. So you get 1 over here, 1 over there, you get 2 cells, right? And basically if we calculate 8:2, 4:1, this becomes your answer. Okay, what will be the ratio of spermatozoa and ova produced at the end of gametogenesis? This is the answer for your question. Fine. Now, the government of India legalized MTP with some strict regulations in the year. In what year did they regularize it? They said legalize it, not regularize it. Legalize it. Any guesses, my dear children? Pretty hard up here. 1951, 1961, 1971, 2001. Your answer is straight from your NCERT. No need of explanation. It is 1971. This is the year when MTP was legalized. Tell with me, 1971. 1971, not 81, not 91, not 61, not 51. This is how they will give you. They will give you 1 at the end for making you confused. Okay, so it is 1971. 1. Remember it. Tell 10,000 times, you will remember it. Okay, at least tell 10,000 times. It's easy for you rather than losing 1 marks in KCET. Yes. Now, 41st question. The process in which a small part of the vas deferens is removed and tied up through a small incision is known as. What are you doing? Vas deferens. You're cutting the vas deferens off. What is vas deferens? It is a accessory duct that carries the sperm. So basically, if you cut it, will the sperm be transported? No, my dear children. It won't be transported. This is known as surgical method of contraception. You're doing surgery, and with the help of this, you are helping with contraception. You're not allowing pregnancy to happen. Why? Because the sperm is not going, my dear children. If sperm itself is not being produced in the semen of the men, my dear children, how can it go fertilize the ovum? Cannot. You get it? And what do you call this process as? Vasectomy. When you are cutting the vas deferens, you call this as vasectomy. Cool? Works? So this becomes your answer. MTP is medical termination of pregnancy, my dear children. That is induced abortion. Yeah, you are inducing the abortion. And tubectomy is when you cut the fallopian tube such that the ovum is not transported now. And gift is gamete intrafallopian transfer. Suppose I am not able to produce gametes. So I am going to take gametes from some other woman and put it inside my body. Not by myself, doctors would be doing it. Okay? Only if I want to conceive. Yes. Now, test cross in pea plant is. What do you mean by test cross? Any guesses? A cross between F2 tall plant and recessive parent, a cross between F2 dwarf plant and recessive parent, a cross between F2 tall plant and dominant parent, a cross between two F1 plants. Can you guess the answer for this question? So, test cross, my dear children, you're testing something. What are you testing? So, you are testing, I have a tall plant, but I don't know, ma'am, whether it is tall like this or is it tall like this. Okay. Then how do you confirm it? You are going to confirm by making it cross with a recessive parent plant. So how do you do it? So for that, you make a cross like this. You write capital T here. You write small t here. So these are the gametes for this plant. These are the gametes for this plant. Similarly, over here also, small t, capital T, small t. Okay, when this and this fuses, you get capital T small t, this and this, capital T small t, this and this, capital T small t, this and this, capital T small t. All of them are tall because at least one allele is tall over here. Okay, so all of them will be tall because that is dominant. So I get all of them are tall. Over here, let's see what happens. Okay, so capital T small t, capital T small t, capital T small t, capital T small t, small t, dwarf, small t, dwarf. So two of them are tall, two of them will be dwarf. So with that I understand that, ma'am, the tall plant that you see, basically it's not like this, it's like this. So you cannot use it for crop breeding programs. Okay, you should take the homozygous one. That will be beneficial. In that case, you will get tall plants later on as well. Okay, this is the reason why we do test cross, for testing what is the genotype. Okay, so phenotypically it will be showing the dominant type. Phenotypically, physical appearance will be that of the dominant trait, which is tallness. But genotypically we have no idea, and for that what you are going to do, you are going to go back and cross it with a recessive parent. You are going to cross it with a recessive parent, and then you see the thing. Okay, so a cross between F2 as in next generation plant, tall plant with recessive parent is the answer. A cross between F2 dwarf plant with recessive parent, you do not need to do that. Okay, a cross between F2 tall plant with dominant parent, again you won't get the, because if dominant parent is there, all the children are going to show the dominant allele. So we do not want that. A cross between two F1 plants, this is the only answer. Remember, you are doing a back cross over here. So all test crosses are back cross because you have to go back. But are all back crosses test cross? Didn't understand? Tell me the answer for this question. Are all back crosses test cross? If you have attended my live, you might be knowing the answer for this. Are all back crosses test cross? Tell me the answer for this question. I will be really, really happy. The genotypic ratio of incomplete dominance is what? What is the genotypic ratio of this? So incomplete dominance you see in Snapdragon, you have capital R and you have smaller. Okay, this is red plant, this is white color flowers. So yes, so the gametes are capital R, smaller and smaller. When you fuse this with this, you get capital R smaller, this with this, capital R smaller, this with this, capital R smaller, this with this, capital R smaller. If you see, all of them has the same genotype. If you talk about the phenotype, all of them are going to be what? Red? No, they are not going to be red because, not dead, not going to be red as well. Why? Because they show incomplete dominance. They are not completely dominant, which is why, my dear children, all of them are going to be pink. In the middle shade, they are going to be what? Pink in color. Here you cannot find genotypic ratio. Let's go to the next level. Okay, this is F1 generation. I am going to cross between F1 now. So RR and capital R, smaller. Capital R, smaller, capital R, smaller. This and this fuses, you get capital R, this and this, capital R, smaller, this and this, capital R, smaller, this and this, smaller. If we see this genotype, capital R, there is only one like that, smaller, there is only one like that, two of them are capital R, smaller, so there are two. Okay, genotypic ratio is what? 1:2, 2:1. If you are asked about the phenotypic ratio, how do you answer it? One is red, ma'am, one is white, over here two of them are pink. Phenotypic ratio is also 1:2:1. Got it? Now I hope it's clear. So, yes. Now, question number 44. Find the incorrect statement among the following. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to some of male progeny. In sex-linked recessive trait means basically, suppose, okay, this gene is affected. Will this person have the disease? No. Why? Because it's a recessive trait. Both of them should be like this, then only it will be expressed. Okay, now the question is, basically, in a sex-linked recessive trait, the gene is transmitted from unaffected carrier mother. So, she will be just a carrier, she won't be having the disease unless and until both of them are affected. Unaffected carrier female is passing this to some of the male progeny. Is that so? Let's see if it's that so. So, basically the father is X and Y, suppose he is not affected, okay. Anyways, so from here you get X, here you get Y. So, here you get a child who will be having the disease. Why? Why this particular child will be having the disease? Because in males there is no other X chromosome to show the effect, which is why the males are going to be affected if they have at least one X chromosome that is affected. So, this guy is affected. But what if this X chromosome came and this Y came? Then it's not affected. So, can I say some of the male progeny are going to be affected? Yes, only some, not all of them, right? So, this is correct. Okay, this is correct. What we have to find out? We have to find what is incorrect. Now, accumulation of phenylpyruvic acid in brain results in mental retardation. Do you think phenylpyruvic acid, when it accumulates, it causes mental retardation? Yes. And in which disease do you find it? Phenylketonuria. Okay, phenylketonuria is when you have a gene that is known as, say, a gene that codes for an enzyme that is phenylalanine hydroxylase. So, this enzyme is not produced, which is why phenylalanine, which is an amino acid, it stays in our body, okay, rather than getting converted into tyrosine. So, it does not get converted into tyrosine, it stays in our body in the form of phenylpyruvic acid and causes mental retardation. So, this is also correct. Individuals affected by Down syndrome will have congenital heart defect and are more intelligent. Okay, they have congenital heart defect for sure. But are they more intelligent? No, my dear children. Here also in Down syndrome, since they have one extra chromosome, that is 21st chromosome, it changes a huge lot of things and they would also be not mentally really strong. So, it is wrong. It is not more intelligent, not mentally strong, as in they would be having mental retardation as well. So, this is a wrong statement and that is what we had to find out. We had to find the incorrect statement. Now, Turner syndrome is caused due to the absence of one X chromosome. Is it? Yes. In Turner syndrome, you just have one X chromosome, there is no other X chromosome coming, which is why it is known as XO condition. This is what? XO condition. So, the only incorrect statement that you have is C. Is what? C. Cool? Now, in a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled green seeded plant, okay. Okay, so you have round and yellow, true breeding, as in both of them are homozygous, yeah, and then you have what? Wrinkled green, so it will look like this, okay. Seeded plant, the ratio of segregation of round and wrinkled seeded trait is what? The ratio of segregation of round and wrinkled seed traits is what? You are asked not about any dihybrid things. You are just asked about what is the ratio of this and this, round and wrinkled. So, you have to go back to the monohybrid ratio. In monohybrid ratio in F2 generation, Okay, in F2 generation, what is it? What is it, my dear children? So in F1 generation, my dear children, capital R, small r, smaller. I'm segregating all the things because you're not asked about anything else, right? So here all of them, my dear children, will be capital R, smaller, capital R, smaller, capital R, smaller. This is F1 generation. F2 generation is where capital R, small r, basically the progenies are being, pollinated. Over here, capital R, small r. Capital R, smaller. This and this goes, you get capital R. This and this goes, you get capital R, smaller. This and this, capital R, smaller. This and this, smaller. So what is the ratio? If you are asked about the ratio of segregation of round and wrinkled seeds, you see 3 of them are going to be round and 1 of them is going to be wrinkled. So 3:1 is something that you have to find out. Remember, in the question what is asked, you have to try to find that. Okay, not what you know about it. Fine. So in a dihybrid cross, they started with a dihybrid cross just to confuse you guys. So they took 2 characters at a time, round and yellow and wrinkled and green, but then they asked about the ratio of segregation of round and wrinkled. So you have to go back to the monohybrid cross. Remember tall and dwarf plants? So that is 3:1. That is what? 3:1. If you are asked about the genotypic ratio, what will you say? It is 1:2:1. Okay, here you are asked about the phenotypic ratio. So I hope, children, you are able to understand what I am trying to tell you. Now going ahead. Yes. Stanley Miller stimulated the conditions of prebiotic earth using spark discharge apparatus. Which organic compounds were observed by him on analyzing the end product of his experiment? So Stanley Miller did the experiment, my dear children. So he observed, he took all the inorganic things. For example, he had methane, that is CH4, ammonia, that is NH3, listen to me carefully, and H2, and also water vapor at a very reducing atmosphere, that is where there is no oxygen, and also the temperature was 800 degrees Celsius. Now what was the end product of it? From these inorganic things, you already got some organic stuff. What was this organic stuff? Was it pigment? Was it fat? Was it nitrogen bases, or was it amino acid? The answer for this question is what? Amino acid, my dear children. It will be what? Amino acids. Amino acids were the first thing that you got. Remember that. Okay, and please remember these guys also, CH4, NH3, this and water vapor, and then what? Temperature is 800 degrees Celsius and it is reducing atmosphere, that means no oxygen. Okay, going ahead to the next question. Most ape-like ancestral primate was? So we had the ape-like, so starting off with the evolution of man, we have 2 guys, okay, Dryopithecus and Ramapithecus. Ram, it suggests like it's man-like, right? So Ramapithecus were more man-like. Dryopithecus, they had dried scaly body, they were itching, it's like chimpanzees, like ape, right things. So basically, Dryopithecus were the ape-like ancestral primates that we had. Remember that, okay? Who? Dryopithecus, not Ramapithecus. So A becomes your answer. Ramapithecus, man-like. From Ramapithecus came Australopithecus. From Australopithecus came Homo habilis. The first hominid features, human-like features were found in Homo habilis, Habibi. Yeah, and then Homo erectus. From Homo erectus came Neanderthal man, and also came Homo sapiens, which is the last thing that to come. Fine. Now, the principle of vaccination is based on which property of immune system? What property of immune system is being taken care of in vaccination, my dear children? So we were all given vaccination for COVID, right? You remember that era? So what was it basically depending upon? What is the principle behind it? So the thing is, basically whenever we give antigen in our body, whenever we have an antigen inside our body, a foreign particle inside our body, our body produces what? Antibodies against it. Now we cannot wait for that antigen to come and affect our body and then to produce antibodies, because in that happens, my dear children, we are going to get affected. Our body at first, you call it as primary response. The primary response is going to be weak. It is very new for our body. The thing that is coming in is very new for our body, and our body might take some time to produce antibodies, and while this duration we might also die. Okay, so instead of waiting for that particular point of time, the scientists thought that why not give a demo to our body. So what they did, scientists did was so they took some weakened pathogens or the parts of the pathogen. not exactly the whole pathogen, either the part of the pathogen that can ignite an immune response in us or probably a weakened pathogen. They put it inside our body such that our body produces certain antibodies against it. Okay, just give me a second. Ma'am, there are 12 questions. Can I complete it? Okay. 10 minutes.
12:45.
Yes. So, where were we? Yeah. So, basically, we are putting inside our body. Yeah. And our body are going to produce two things. One is antibodies against it. Yeah. And the second thing is memory cells. Why memory cells? Because our body need to have a memory of, yes, this has happened and next time it can happen, and this time I have to produce these antibodies, my dear children. So, basically, just for having a memory of this particular incident that have taken place in our body, we do vaccination. Okay, so that our body could be producing antibodies in a faster rate. The secondary immune response would be obviously faster because the memory cells are being produced. So, to ignite this, we have memory cells. Works? Yes. Now, question number 49. Genome of HIV replicates in the macrophages with the help of an enzyme called what? Genome of HIV. HIV has a genome. What is it? It is a RNA. It is what? RNA. Now, this RNA, the whole plan of HIV is basically to get inside our nucleus and get integrated into our DNA. But can RNA integrate into our DNA? No. So, what happens is that this RNA is made into a DNA. How? How it is made into a DNA? Basically, what do you call this process of making a DNA to an RNA? It is known as transcription. Now, we are doing ulta of it. So, it is known as reverse transcription. Reverse transcription. And the enzyme which is there, it is known as reverse transcriptase. It is known as reverse transcriptase. So, C becomes your answer. Not DNA polymerase, not RNA polymerase, but reverse transcriptase. Okay? Works? Going ahead. Read the following statement. Statement 1, and then you have what? You have statement 2 as well. Okay. Now read the statement. Statement 1 says morphine is obtained by acetylation of heroin. Wrong. Heroin is obtained by acetylation of morphine. So this is a wrong statement. So you might remember just acetylation, but you might get things wrong. Remember, morphine is not obtained. Heroin is obtained from acetylation of morphine. Okay, wrong statement. Statement number 2, cannabinoids are known for their effect on cardiovascular system. Is that true? Yes, they have an effect on our cardiovascular system, my dear children. If we talk about coca alkaloids, they have an effect on our CNS, central nervous system. They mess with the dopamine receptors, okay, that is cocaine and things like that. If we talk about opiates, opium poppy and things like that, so such as heroin and what do you call morphine, okay, they are going to affect our CNS again and also our gastrointestinal tract. Okay, so starting with your opiates, they are going to affect your CNS and also your gastrointestinal tract. Cannabinoids are going to affect your cardiovascular system, and coming back to CNS, we have coca alkaloids or cocaine and things like that. Okay, please remember it. So, what is the answer? This is correct, this is false. Statement 1 is false and statement 2 is correct. This becomes your answer for the question. Next question. Mule is the result of what? Mule is what? When a donkey and a horse is being crossed, you get mule. Is mule fertile, my dear children? Can it produce offsprings? No, they are sterile. They are what? Sterile. Why are they sterile? Because you are crossing between two species. You are crossing between two species, which is why the product is would be what? Sterile. So this is known as interspecific hybridization. Between two species you are crossing, that is why it is known as interspecific hybridization. Cool? It's not crossbreeding, it's not outbreeding, it's not outcrossing. First of all, this particular portion is not there in your course also. So your answer is interspecific hybridization. Now, identify the bacterial disease among the following. Brown rust of wheat, tobacco mosaic disease, tobacco mosaic virus, okay, that causes the disease. Now, black rot of crucifers and late blight of potato. What is the answer for the bacterial disease, my dear children? The answer is black rot of crucifers. This is a direct question. Okay, black rot of crucifers caused by a bacteria. Works. Okay, going ahead. Match the nutrients given in list 1 with the source in list 2. Okay, fine. Vitamin A. So we say vitamin A is there for improvement of your eyesight, and for eyesight basically you should be having carrot. Okay, carrot, the orange color thing. So vitamin A is found in the orange color stuffs, my dear children. So what is orange is over here? Only carrot. So vitamin A, can I link it with carrot? Can I link it with carrot? If you're not able to see, I'll zoom it in and show it to you. Okay, so 1 goes with R, whatever options do have 1 with R. Only 1, and with that I get the answer. 1 goes with R, 2 with S. Let's see. 2 is what? Single cell protein. Okay, a single cell can give rise to a whole bunch of protein. So 2 with S, spirulina. Yes, my dear children, it's a unicellular thing and it has all the essential amino acids in there, which is why you call it single cell protein. Okay, spirulina species. Now vitamin C. Vitamin C you can find in bitter gourd. Bitter gourd, no one likes it, but it has enough amount of vitamin C, very good for your health. Okay, now you have protein. Beans are the ones that gives you proteins. So hence what will be your answer? Hence what will be your answer? Single cell protein is S, vitamin C is basically bitter gourd and protein is beans. So hence your answer becomes 1 is R and 2 is S and 3 is P and 4 is Q. This becomes your answer. Works? Chalo, I'm keeping the promise given to Aishwarya ma'am. Yes, so that she gets the studio. Now, the chemical substance which are produced by some microbes which can kill or retard the growth of other microbes are known as. For example, there is a microbe, okay, and I am going to release something which does not allow the growth of another microbes. What is this something that I am going to release? This is statins, streptokinase, cyclosporins, antibiotics. What is the answer? It is antibiotics. I am going to produce antibiotics which does not allow the growth of another organisms out there. Statin is what? Statin is a blood cholesterol lowering agent. Write it somewhere. Blood cholesterol lowering agent. Streptokinase is a clot buster. It bursts down the clot. Okay, kinase. Something it is going to cleave something. Cyclosporin A, my dear children, it is a immunosuppressive agent. For example, I get a graft, okay, from some other organism, I get kidney, not other organism, other human being, I get kidney. Now, my body will react to it because it's foreign. So, to make my body suppressed, okay, immune system a bit suppressed, I give immunosuppression. So, cyclosporin is going to act like an immunosuppressant. The answer is antibiotic. Cool? Next, select the correct statement from the following. Methanobacterium is an aerobic bacterium found in the rumen of the cattle. In the cattle, do you have some bacteria? In the rumen of the cattle, do you have some bacteria? Yes, there is methanobacterium, my dear children. Sure, for sure. But is it an aerobic bacteria? Does it need oxygen? No, it is an anaerobic bacteria. It is what? Anaerobic bacteria, my dear children. Why do you think methanobacterium is there in the rumen of the cattle? Because, my dear children, cattle have plants and plant cell wall is made up of cellulose. And for the breakdown of cellulose, for efficient breakdown of cellulose, you need the help of methanobacterium. It is not the one that directly breaks down cellulose. Please remember that. It helps. Okay, but it's an anaerobic bacteria, so this will be a wrong statement. Biogas is produced by the activity of aerobic bacteria. Wrong. It is produced by the activity of these methanobacteriums, methanogens, my dear children, which are anaerobic. So again wrong. Biogas is pure methane. Wrong. Biogas is not pure methane. It is H2S, it is CO2, it is methane, all of it together. Okay, remember it's not just methane. Now, activated sludge in sediment tanks is a rich source of aerobic bacteria. Is it the activated sludge? Activated sludge is basically, okay, happens. Listen, activated sludge is basically when you have, okay, so you have a container, you did the primary treatment, you filtered things out using filtration. All the things that were floating, you removed it. All the things that were sedimenting, you removed it. Now, whatever you have for treatment, it is known as primary effluent. Okay, this primary effluent, I am going to put it in a tank. Okay, that is aeration tank, and here the aerobic bacteria is going to feed on this organic matter which is there in it and try to break it down. It is trying to cleanse this. Okay, but while it does so, it becomes friends with, aerobic bacteria becomes friends with some fungal filaments and forms flocks and things like that. But anyways, it doesn't work, it breaks down the organic matter up there. Now what happens, since it is broken down, we pass the whole thing to a next thing that is known as settling tank. Okay, or a sedimented tank. What happens over there? Basically, the aerobic bacteria and the fungal filaments, they settle down, and whatever is left behind, my dear children, it's great because the organic matter, everything is broken down. It's good to go into the rivers, so it is put down to the rivers. Whatever is left behind is what? Activated sludge. And activated sludge, as I said, what is settling down? The aerobic bacteria and the fungal filaments. So activated sludge in the sediment tanks is rich, a source of aerobic bacteria. Correct. It is what? A correct statement. Okay, now just give me a second. I'll be right back. I have to close this and come back again because of my hair. Okay, so your answer becomes D. This is the only correct statement out there. Fine. Next question. Which of these enzymes is required to cleave a plasmid? A plasmid is what? Extra chromosomal double-stranded DNA. That is, apart from the chromosomal DNA, we have some extra thing that is known as extra chromosomal double-stranded DNA, which is plasmid. Now, for cutting plasmid, do you need ligase? Ligase is for joining. Do you need endonuclease? Yes, endonuclease cleaves from between, from inside. So, yes, you need it. Exonuclease, exonuclease cuts from the ends. So, there is no end, it's circular thing, right? So, not exonuclease, it is endonuclease. Polymerase, no. Polymerase you need for replication and things like that. So, it is just endonuclease. Restriction enzymes, you have two types, exonuclease, endonuclease. Endonuclease is the one that cut from the middle. Exonuclease cuts from the ends. We do not have end over here, so it will be endonuclease. Now, the natural reservoir of phosphorus is, what is a natural reservoir of phosphorus? Where do you find most amount of phosphorus? Rocks, soil solution, detritus, atmosphere. Detritus is basically the dead and decaying things that we have. What is the natural reservoir of phosphorus? Should I leave this question for you? Should I do it? I'll do it. It is rocks. My dear children, my dear little champions, it is what? Rocks. Rocks has enough amount of phosphorus. Please remember that. Okay, and phosphorus does not have gaseous cycle. That also you have to remember. 58th question. The sequence of communities of primary succession in water. In water, you have phytoplanktons, my dear children, for sure. So now all of them have phytoplanktons except this guy. As the initial one, there is no phytoplankton, right? So phytoplanktons are the first one to come in, and then only came the other organisms. So phytoplanktons came in, then I'll tell you what came in. There were rooted hydrophytes, as in they were having roots. Slowly, they were free-floating hydrophytes. Okay, they started floating. Now there were reed swamps, then there was marsh meadows, and then the scrubs and the trees. So what becomes your answer? D becomes your answer. So over here, phytoplanktons and directly scrubs, wrong. Phytoplanktons, free-floating, where is the rooted ones? Wrong. So first thing that you need, phytoplanktons, then you need rooted hydrophytes, and then the one lives in water. Okay, then free-floating hydrophytes, then reed swamps, then marsh meadows, scrubs, and then trees. This becomes your answer. D is the answer for your question. Now, 59th question. Match the type of adaptation given in list 1 with their examples given in list 2. Select the option showing the correct combination. Okay, biochemical adaptation, behavioral adaptation, physiological adaptation, morphological adaptation. So, physiological adaptation is done by kangaroo rats. I'm pretty sure rats. Why? Because basically they have very concentrated urine. So, physiologically they are adapted, my dear children, right? Can I say that? Yes, I can say that. So, 3 goes with S, and with that can I find the answer? I have 2 options with 3 with S. I have 2 options with 3 with S. Now, anything else that you have? Biochemical adaptation, behavioral adaptation, morphological adaptation. Behavioral adaptation, desert lizards, my dear children. They have behavioral adaptation to live there. So, can I say 2 goes with P? 2 goes with P. So, what was the first thing? 3 goes with S. 3 goes with S. I had 2 options like that. So, I had to cancel these options. I didn't cancel. Okay. Now, 2 behavioral adaptation goes with desert lizards. To 2 goes with P. I have this, and this becomes my whole answer. Let's see. 1 goes biochemical. Biochemical adaptation goes with Q. Deep sea fishes, yes, true. Okay, and then you have morphological adaptation. They have the thorns and stuff like that, which decreases the surface area for exchange of, for less amount of transpiration to happen. Okay, instead of having a big leaf out there, they have the leaf being formed into thorns. There is no much amount of transpiration happening because obviously they need water. They are desert plants. So this is correct. Okay, fine. This becomes your answer. Last but not the least, this is a direct question from your NCERT. The annual net primary productivity of biosphere is approximately what? Net primary productivity, the whole productivity that you have of the biosphere is what? 170 billion tons, 55 billion tons, 170 million tons, or 55 million tons. What would be the answer for this particular question? The annual net primary productivity of biosphere. Can you try to find the answer for this question? It's very, 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 very important. Any guesses? Any guesses? So your answer is what? 170 billion or 170 million? This one. Okay? Just give me a second. Okay? Now the next question, The annual net primary productivity of biosphere is approximately what? 170 billion tons, 55 billion tons, 170 million tons and 55 million tons. Very important question straight from your NCERT. Net primary productivity, as in the whole thing, after including, for example, I am an organism, okay, I have the food being produced, I am using it for myself, and then whatever is left behind is known as net primary productivity. So over here, whatever is left behind in the whole biosphere, my dear children, it is 170 billion tons. This is what? 170 billion tons. This should be your answer. 170 billion tons. And with that, we finish all the questions, 60 questions of your previous year paper. My dear children, I hope it was helpful. And please, if you have got any answers wrong, write in the error book. Make sure you're writing in the error book. I'm telling you, this step is very much important, and rectify it. Go through it once more. You will not repeat the same mistake again. I'm pretty sure about that. Yes, so take out bit amount of time for that. That's all, and wishing you all the best. If you like the video, do not forget to like. Okay, click on the like button and also comment down, and also subscribe to our channel. Also share it with your friends who are in need. Wishing you all the best. Take care of yourself. Bye bye. Take care.